If you're watching this video for any kind of points or credit, we're going to do it as we usually do, meaning that throughout the video, I'll give a list of attendance words or attendance phrases. You're going to want to write those down. Also, when I give an attendance word or an attendance phrase, I want to take a screenshot of the screen. And then at the end of the video, it's all said and done, send me an email, one email per video, please. In the email, include the date of the video, but then also include the list of attendance words and their accompanying screenshots. And then finally, one more thing that you're going to need is to ask one question about the material cover. That's it. I will go ahead and say that the first word for attendance is going to be open. So you should write that down. Just take a screenshot, and when you send the email with all the words, one of them should include the word open, and one of the screenshots would be one of me pointing at the word open. And that's it. Like I did mention, I think it's the first time I've reiterated this. Well, maybe not the first time. It's been a while, so it's a good reminder. When you send these emails to claim the extra credit, like I said, you need to mention what the date is. Like, what is the date on the video? So today's date will be, as you can see up here, 2/18/26. Now, granted, when you're sending the email and it has the screenshot, at least in this particular example, it would include that, but it's not always the case. So make sure you include that either in the email or the subject line. That's it. It is about 7:40. So you can skip ahead about, 15 minutes, and that's when the lecture will get started. Morning.
So exam 2 is next week.
No, it'll be a while.
Oh, it is?
Yeah. So you generate about a month between exams. Granted, exam 1 was a little bit behind schedule. So if we stay on schedule for exam 2, it'll be a little bit less than a 1 month gap, but we will see. All right. There it is here. Good morning. It's here. Good morning. Here. All right, we've got less than 3 minutes, so I'll go ahead and start with some of the less important stuff. As I already mentioned, exam one will be open for retake, but that'll start tomorrow. There were some issues, somebody who was sick needs to take it, things like that. And I've had to have taken it by tomorrow. So anyway, it'll open up tomorrow, like I mentioned on Monday. The retake it's untimed, it's unlimited attempts. You're allowed to use your book, things like that. So it can do nothing but help your grade. And when you're done with it, once you're satisfied with your grade, email me, and I'll average your first grade with your highest grade. So again, nothing to lose. Even if you get a lower grade, then just don't email me, and we'll keep your original highest grade. And again, like I said on Monday, this is good because it'll help you, even if you already got a good grade. Not only might it help you grade a little bit, but also to help you prepare for the final exam. The one thing I did mention on Monday, or I don't remember if I did or not, but it closes when exam 2 closes. And Barrett asked a good question. When is that going to be? And it's in there. The due dates are online. I forget when. Generally, there's about a 1 month difference between exams. Now, obviously, since exam 1 was a little bit pushed back a little bit, exam 2 is either going to be a little bit earlier than that gap, or it'll also need to be pushed back. But we'll see. And it'll be like that for every exam this semester, except the final exam. The final exam will be on that Monday at 8 a.m. in this room. That's kind of not flexible. It's not up to me. But anyway, yeah. So any questions about exam 1? Retake. So again, nothing new there. Just basically reiterating what I already said on Monday. Pre-BAP 6, just to remind you, is due on Friday. This is an important one. This is about photosynthesis and respiration. This is the important lab because you have a lab report for it. This is the one lab that you have to do a lab report. That's also worth 20 points. So for that reason, academically, it's more important than the rest. So be prepared. You're going to have 2 sets of video questions, as you're probably used to already. Then you have an introduction that you need to read with a set of questions to make sure you understood the introduction. And finally, don't mess this up. You're going to have to move forward and read the whole lab like you always should anyway. But in this case, you're going to put your 5 hypotheses. It's going to ask you, like, hey, what do you think is going to happen? Do you think light intensity is going to affect respiration or not, or photosynthesis or not? But also be aware of this. These hypothesis questions, I think maybe for the first time, ask you for your reasoning too. So it doesn't just ask for your hypothesis, but it's also like, hey, why do you think that? And it seems like a pain, but this is all to help protect you, or not. I saw the mask about protect. Very smart. This is all to help prepare you for your lab report. Because when you write your lab report, it's not just you don't just have to write down your hypothesis. You also have to write down why, your reasoning for your hypothesis. So this lab worksheet. It's going to be slightly different than the ones you're used to. But again, it's there to help prepare you to write your lab report. And a lot of the stuff, when you write your lab report, you'll be able to just copy it from your lab worksheet and paste it to your lab report. So any questions about the pre-lab, let's do this Friday. All right. And as always, is there a question? Okay. As always, the pre-lab, so you don't have to do them alone. I'm more than happy to meet you online and help you do it. And I don't know if I've reiterated this enough with you guys, but I have with my online students. From now on, well, I guess this will be a little bit different, but so I'm offering them 20 lab extra credit points if they meet me online to do their labs, even if they don't need my help, if I'll just be there if they need me. But also I'll give them a little additional extra credit points if they can maybe give me some feedback, because I want these lab worksheets to be as useful and helpful for you as possible. but I can't do that without doing pre-lab. So anyway, yeah, if you meet me online to do your pre-labs or your labs for that matter, you'll get at least 20 points of extra credit, lab extra credit. But also I might give you a little bit more if you kind of talk to me like, hey, that's not, you should maybe reword that. That's not clear. Or maybe you should put this there. We can talk about it. Maybe I can say, well, I can't. Here's the reason why I do that. But it'll be very useful for everybody involved. Anyway, today I've already put this in writing in the lab announcements, the course announcements, but we're going to meet here for lab. We're not doing any experiments. It's just going to be a pre-lab lecture, much like you're getting a lecture right now. And just like you're getting the lecture right now, the same thing for lab. You're going to write down the attendance words. You're going to send an email with the attendance words and one question. And that's how you're going to get your 20 points for today's lab. No experiments, no lab worksheets, just pay attention to the lecture and that's it. So any questions about that? All right, so speaking of which, I don't even need to say this again. Same thing, participation, attendance, it's the way it always is. Send the email in. If you're here or online, make sure you send it in before 9 a.m. If you're watching the video, send it whenever, but make sure to include your screenshots if you're watching the video. So really quick for attendance. Ashley, here. Barrett is here. Let's see what this is about. Of course, if you're online and I say you're not here, don't worry about it. I'm just physically here. Anyway, Wooten is not here. Trahan is not here. Dickens is not here. Clark is not here. Inca is here. Isaiah is not. It's not Isaiah. It was dark. Couldn't tell. It's dark back there. Copes is not here. Smith is not physically here. Watson is not here. Haley is not here. Jake, Lucas is not here. Schaefer is here. Noah is here. Savannah is here. Mia is not here. Edwards, I think, is online, but not here physically. And Wilkes is not here. Any questions? Did I miss anybody? All right, let's get it closed and get it started. So we left off talking about enzymes. Enzymes are very important because, like I said, everything we're going to talk about, at least for the first half of the semester, but all these different processes like photosynthesis, and respiration, and mitosis, and meiosis, all these different cellular processes that you're going to learn, it all happens because of enzyme. These enzymes, there's a very specific enzyme for each specific chemical reaction. So that's what we just finished talking about. We finished talking about how basically how enzymes work. But they get a little bit more complicated than what we left off. So let's talk about this. Actually, before we do that, the first word for attendance, at least for you guys, if you're watching the video, I already gave you the first word. But for you guys, the first word for attendance will be plug. Word plug. Anyway, so you know the basics of how enzymes work. You know that it's all about their shape, and the shape is very sensitive to their environment. You know that they have a very specific shape in their active site, which their very specific shaped substrate attaches to, and that's where the magic happens, so to speak. So that in mind, I can teach you about something called enzyme inhibitors. So think about what the word inhibit means. Like if you're not in biology class and someone said something like, yeah, we went to a party last night, a friend drank too much and they were really uninhibited, right? In that case, that means probably because they had too much ethanol. The thing inside them that keeps them from doing something that they shouldn't be doing was not there, right? So an inhibitor basically stops something from happening. And that non-biology example, your inhibitions keep you from doing something dumb, like maybe confessing your love to your best friend's girlfriend or something like that. But that's what an inhibitor is. So an enzyme inhibitor, well, it just does exactly what an inhibitor does. It stops something from happening. Specifically, it stops an enzyme from doing what an enzyme does. And it does that, and there's different types of enzyme inhibitors, but either way, either type we're talking about, they all work in the same basic way, which is they're going to bind to the enzyme in some certain spot. And by doing that, that's going to disrupt the function of the enzyme. And there's going to be two of them. So before I move forward, let's let you first understand that this is how both the different types basically work. And then we'll get into the two specific types. Of course, you can see one of them right now, but yeah, the first one is called competitive inhibition. That little meme I just found somewhere randomly is a great example of competitive inhibition. So here you have this enzyme, right, with this very specific shape. You have that substrate that has also a very specific shape where it fits into the enzyme's active site. But in this case, there was an inhibitor. Notice the inhibitor doesn't have the same exact shape as the inhibitor, doesn't have the same exact shape as the substrate, but it doesn't need to have the same exact shape as long as it's close enough, right? It's sure it hasn't blocked the whole thing, but it blocked that part. So if the substrate can't completely fit into the enzyme's active site, then it's not going to be able to do its job. So, whoever drew this little cartoon happened to draw the inhibitor there. Of course, it could have drawn it there, or it could have drawn it where it completely matched the shape of the enzyme substrate. But yeah, so that's competitive inhibition. It's competing against the substrate. In a sense, it really is competing. You can almost think of it as a race, right? All these things just float around in solution, and they're competing to see which one can get in there first. And if the inhibitor gets in there first, that's going to block the substrate competing. So, are there any questions about competitive inhibition? All right. Before we move forward for a little bit of extra participation points, can anyone guess what the next type of inhibition is? By using some good guessing. If this one's called competitive inhibition, any guesses what the next one might be called? No? All right. Non-competitive inhibition. So again, we're still binding, like I said at the beginning, these things are still binding to the enzyme, but in this case, it's not binding to the active site. It's just going to bind somewhere else. So how does that work, right? How's that going to keep it from doing its job? Well, this is where it gets very slightly trickier, not much, but you need to know this. When it binds to wherever it is on the enzyme other than the active site, it also changes the enzyme's shape in some way. And as you know, it's all about the shape when we're talking about these enzymes. So because of that, the active site can no longer accept the substrate. So it's not blocking the substrate like the last one we talked about. It's just changing the shape of the active site so the substrate can no longer fit. To make things a little bit more complicated, another word for non-competitive inhibition is allosteric inhibition. I'm not sure which one of those I use on the exam. Currently, I plan to use both. I need to go in there and look at the exam and see what I have written. But hopefully, if I have a question about that, it already says non-competitive / allosteric inhibition. Are there any questions about this slide? The next slide I'm going to show you, I'll give you a couple seconds before I go there, but the next slide is going to be one image that shows the different types of inhibition and one enzyme working normally. And the image I'm going to show you is not from your textbook, it's from an old textbook. There you go. Everybody good? I'm not going too quick, right? Remember, feel free to slow me down or speed me up. Anyway, see what we can see at the top right? We have a square-shaped enzyme. It's got a round-shaped active site because it has a round-shaped substrate, and also has this little notch. We won't even explain that yet in that first picture, right? So that up there at the top is that square-shaped enzyme ready to do its job, and the substrate comes in and it's doing its job, whatever that job may be. So making a new, or making some sort of new product in that chemical reaction. We just don't know what. Here we have competitive inhibition. As a matter of fact, your old textbook didn't even get into the difference between competitive and non-competitive, which is why it's labeled the way it is. But yeah, competitive inhibition, right? Here we have the same enzyme. We have the substrate, the inhibitor, which again, it has the right shape to fit into the active site, which blocks the substrate, which blocks it from doing its job. And then finally, here at the bottom, we have an example of non-competitive inhibition or allosteric inhibition, where the active site is left alone, nothing's binding to it, but the inhibitor binds somewhere else on the enzymes, which causes that, in this particular case, causes it to shrink, right? It was wide open, but it's kind of caused it to change its shape and get smaller. Again, this is just an example, right? All enzymes have different shapes. All substrates have different shapes. So when I say the non-competitive inhibitor changes its shape, it could be some other example, right? Not all of them look like that. As a matter of fact, so far, the other examples I've given you have been little hexagons. When we talked about lactose, what was the other one? It doesn't matter. Anyway, the point is, these are just examples. So don't think all enzymes have that shape and all substrates have that shape. They're all very specific. Are there any questions about this slide? Okay, here's some concepts I've mentioned earlier. Now we're going to dive into them a little deeper. So again, we're still talking about how enzymes work. You've already learned the basics. Now we're still learning some of these exceptions and these add-ons, if you will. Sometimes enzymes need help. Sometimes this help comes from temporary bonds between other molecules. Sometimes it's permanent. And it gets its help from things called cofactors and coenzymes. If there's any questions about this on the exam, they will be few. And I don't currently plan to have you go so deep as to where you have to distinguish between cofactors and coenzymes. Like, not necessarily, like, in my mind, you should just be able to think, all right, enzymes need help, and they need help from cofactors and coenzymes. Now I'm going to tell you what cofactors and coenzymes do, but I don't feel like for the exam I'm going to ask you which one of these is done by the cofactors and which one of these is done by the enzymes. But anyway, cofactors, these are inorganic ions. Like iron is a great example. Because hemoglobin, right? You have a hemoglobin, that's a protein. Well, that's a pretty good example. Anyway, you have this protein called hemoglobin in your red blood cells, and that's how you carry oxygen. But it needs that iron in the middle of the protein for it to work. It doesn't work without it. So for extra credit, not on this email, but any other email, if you want to look up some examples. What are some examples of how iron is used as cofactors? What are some examples of how magnesium is used as a cofactor? What are some other inorganic ions that are used as cofactors? And that brings us to coenzymes. And this one's slightly more important as far as we're concerned in this course, because later, one of the enzymes I'm going to teach you about has a coenzyme, and I'm going to mention it. So that's nice that now you'll know what a coenzyme is before we get there. But beyond that, coenzymes academically are no more important than cofactors. Anyway, coenzymes are organic molecules, and remember where you learned about that in chapter 2. So these are carbon and hydrogen based. So that's it, basically. Anyway, whether it's a cofactor or it's a coenzyme, both of them participate in reactions without being altered. So they're recycled and used, just like an enzyme, right? So cofactors and coenzymes are not used up in the chemical reactions. Speaking of which, just to remind you, like we said on Monday, in the exam, I'll give you some hypothetical fake chemical reaction and say which one of these is the enzyme, which one of these is the substrate. And I gave you some hints on how to figure that out. So just to let you know, this will not factor into those questions. So when you have, it'll be very basic questions when I give you those questions. I'm not going to give you a chemical equation and ask you anything about a cofactor or a coenzyme or an inhibitor. Now, you might get a question where I ask what is an inhibitor, or ask what do cofactors and coenzymes do. Of course, it's in the name of the coenzyme, right? It helps in the enzymes. Anyway, vitamins are often served as the source of coenzymes. Your vitamin A, vitamin C, that's very often what it's used for. Again, that's very basic. Feel free to look at it for extra credit. Which vitamins and which enzymes do they work with? And are there any vitamins that are not used as cofactors? All of that you can look up for extra credit. And I'd say extra credit because you don't need to know it. If you needed to know it, I would tell you. But feel free to look it up for extra credit. Any questions about this slide? Oh man, this slide was out of order. I apologize. It's not going to matter too much. We were talking about inhibition. There were some more things I wanted to teach you about inhibition. I apologize for this mistake if I can remember. Actually, the next word for attendance is not a word at all. It's a phrase. And it's basically saying something like, remind me to fix the order of the PowerPoint. However you want to word that. If you're all emailing me, then that's going to be like, oh yeah, and I'll go in there and fix it. So how do you want to word it? Remind me to fix the order of the PowerPoint, please. Of course, now that I've gone through all that, I'll probably remember it on my own. But anyway, going back to inhibition, and I apologize, but there's a particular type of inhibition called feedback inhibition. And this is where the inhibitor itself is the product of the chemical reaction. which should make sense. Because think about it, your cells are not finite, right? They have resources and energy, just like anything else in the world, and they have to be very careful how they spend their resources and their energy. Because otherwise they will run out of resources or run out of energy. I use a dumb example every semester. I keep using it. I don't know if you guys are young enough or I guess old enough. I don't know if you're old enough to remember when fidget spinners were a thing. Like a huge craze. I remember like every kid had them for like 6 months there. But anyway, imagine you own a factory, a toy factory. You've got a finite amount of money and a finite amount of resources and a finite amount of employees. And then the fidget spinners kick off. Like, whoa, we got to make some changes. We need to stop making what we're making right now and start making more of these fidget spinners because they're hot, right? And then eventually, once they stop being hot, then they need to adjust again. Like, whoa, we're making too many fidget spinners. We need to change this, change our process. That's basically what this is. Your cells need to make a particular product. And this is a really complicated picture. I don't remember if it's from your textbook or not. But anyway, it doesn't even have to be this complicated. The point is, you have an enzyme, substrate, enzyme 1, intermediate substrate. Yeah, that's pretty cool. I need to find a better picture. Just think about an enzyme, right? It's producing something. But in this case, if it produces enough of it, that thing is going to come back and it will attach back to the enzyme and inhibit it from making any more. So that weird toy example I gave, if it was possible, if somehow, if you had like too many fidget spinners coming out of the machine and one fell back into the machine and stopped it from working, that would be an example of feedback inhibition. So again, once your cell makes too much of something, once it has enough of it, then it'll slow down that process or maybe even stop it. So keep in mind, there's obviously more than one enzyme. So whatever enzyme I'm talking about here, there's multiple copies of them. Yeah, so that's how that works. That is feedback inhibition. So again, the big concept there is feedback inhibition uses the product to basically inhibit the process from happening. So any questions about feedback inhibitions or enzymes in general? Because we are now done with enzymes. Okay. So we're about to jump into next is like the meat and potatoes as far as the exam is concerned. So this is everything we've talked about starting on Monday to now. There'll be a few questions, but not much. You do need to understand the concepts that I've taught you to understand what's coming next. But as far as the exam is concerned, it's not going to be many questions about that material. What we're moving into, there's going to be a lot of questions about it. So what we're about to talk about, something called mostly, here, is going to be cellular respiration. About half of exam 2 is going to be about cellular respiration. And about the other half is going to be about photosynthesis. And there will be some other stuff sprinkled in, like I talked about, enzymes, fermentation, and things like that. So again, what we're about to talk about is very important. And I can go ahead and tell you now, just so you can keep this in the back of your mind as I'm teaching it. For the exam, right, so you can see right here, cellular respiration is made up of glycolysis, citric acid, and oxidative phosphorylation. We're going to learn about that. But the point is, it's made up of 3 parts. So throughout that, for the exam, most of the questions are going to be about, like, all right, which of the 3 parts uses oxygen? Right? Which of the 3 parts produces carbon dioxide? Which of the 3 parts uses glucose? Which of the 3 parts produces the most ATP? So that's going to be the majority of what these questions are going to be about. Which part, which does this, which does that, which produces this, which uses that. Just keep that in the back of your mind as we're moving forward, because that's how the exam will go. By the end of this section, you should be able to explain how ATP is used by the cell as an energy source. That's actually very important, and that will be a test question. If anything, that's the one thing you should remember for this whole exam 2. That's the biggest piece of information you should know. That cells use ATP as energy, right? That's the biggest thing. If you forget everything else, at least remember that. ATP is used by cells. That's our primary source of energy. And then, yes, you should be able to explain how it's used, but more importantly, the fact that ATP is used as energy. And then you should be able to describe the overall result in terms of molecules produced of the breakdown of glucose by glycolysis. So in other words, and you're going to see this in writing again here soon. In other words, for this bullet point, like I said, there's 3 different stages of cellular respiration. Glycolysis is the first one. And when we're done with whatever, I think it's 4.2, whatever number it is. When we're done with this section, you should understand what we started with glucose and what we ended with. Like, what did we produce? Excuse me, glycolysis. So that being said, let's jump into it. The first thing we're talking about is ATP. So as we already mentioned, enzymes work by reducing or lowering the activation energy, right? So activation energy, to remind you, is that energy that's needed to start the chemical reaction. Whether that chemical reaction puts off energy or takes in energy, you at least need some energy to start it, and that is activation energy. And we said that enzymes work by lowering that. But nonetheless, even though they lower the activation energy, they didn't lower it to zero. So you still need some energy to get these things started. So this is where we get our energy, ATP. And this is directly from your textbook. ATP is used for everything in your cell, as far as we're concerned. There's minor exceptions, but as far as we're concerned, everything that your cell needs energy for, it gets it from ATP. So it's a very huge thing when it comes to life. Without ATP, there is no life. ATP is a small, simple molecule, which I'm not going to ask you about. You don't necessarily need to understand that to understand how anything else works moving forward. It contains potential energy for cellular work. That's not going to be a test question, but you do need to understand that. Specifically, chemical energy, because remember, chemical energy is a type of potential energy. And to remind you, like I said, I'm going to keep saying this a lot, you're going to get so tired of hearing it. However much energy it takes to build a molecule, like ATP, is how much energy is released when you break that molecule down. Therefore, the harder it is to build that molecule, right? The more energy it takes to build it, the more energy will be released when you break it down. Again, very important. It is the primary energy currency in cells. Very, very important. Like I did say, though, there are some exceptions, and for an extra credit, you can look up those exceptions. What's the other energy source? But don't confuse this for the exam. There's a few questions that will be very similar. For example, there'll be one question that says something like, ultimately, all energy on Earth, with a few exceptions, came from what? And that came from the sun. So all that energy that's stored in the bonds of the ATP ultimately came from the sun. We're not talking about ultimately, which is the primary energy currency in cells, it's ATP. Don't get confused with that too on the exam with glucose. Because we need glucose. When we talk about making ATP, for the most part, we always talk about glucose. You start with glucose, you go here, you go here, you go here. But obviously you eat more than glucose. But anyway, this is the way we focus on it. We always say we use glucose to make ATP. So don't let that confuse you. Don't get confused with that on the exam. Again, like we said, it powers the majority of energy requiring cellular reactions. Again, for extra credit, you can look up what the exceptions are. But as far as we're concerned, again, for our level of biology, you can just assume that ATP powers everything in your cells, everything that requires energy. And in a sense, they're basically like rechargeable batteries for cells. And you'll see why when we talk about the very simple ATP cycle. Like it's charged, and then it loses its charge, and then your cells charge it up again, and then it loses its charge, and the cycle continues. But that's one picture of it right down there. Just to remind you, or not remind you, I'll let you know, I put a big X through this to indicate this. I'm not going to ask you the parts of the ATP. We will talk about some of the parts, but never on the exam will I give you this picture and say label it, right? You definitely don't need to remember gamma phosphate group, beta, alpha. You definitely don't need to know the difference between the ribose and the base. That's just how it happens to look. How it happens to look will be slightly important in the upcoming slides, but that specific, that the labeling is not important. Anyway, any questions about this slide? Okay. All right. Again, ATP. That last picture I showed you the structure. Like I said, all those details were not that important. But if we just look at it from the big picture, then the structure is a little bit important. Because like I said, it's a big theme of structure and function of biology. This is a great example of it. Not that I expect you to memorize any of this. Again, this one's labeled too. It's not labeled as much as the other one. But we have this adenine, which is the base. Because what we're dealing with, this is a nucleotide, by the way. I forgot to mention that because it won't be on the exam. But remember, there was 4 different DNA nucleotides, A, T, C, and G.
Well, this is the A. This is an A nucleotide. It just happens to have a lot of phosphate groups attached to it. Nonetheless, point is, I'm not going to ask you the different parts, but I am going to explain something about the different parts so you can understand, not for the exam, but just for your own knowledge, so you can understand why this is such an energy-rich molecule. So what we have here, again, we've got the base, and we have these three little phosphate groups attached to each other. And what you probably don't know, unless you know a lot about chemistry, well, first of all, you can see this, right? Those oxygens are negative. Those have negative charges. So you know they want to repel each other. Those things are negative, so they want to repel each other. Meaning, it's going to take a lot of energy to push those things together because they don't want to be there. Not only that, what you might not know is that oxygen itself is a very negative atom. And you kind of had a hint of that when we talked about polar molecules and water, right? For example, the electrons like to hang out next to the oxygen, not the hydrogen. So it's slightly negative in that sense. And another thing that you haven't learned, that you don't need to know necessarily, the double bonds are also very electronegative. The point I'm making here is these things, these little phosphate groups, are very, very negative, which means they're really going to repel each other, which means it's going to take a lot of energy to squish them together, which means, like I keep saying, however much energy it takes to build something is how much energy is released when you break it, for the most part. That means these things have a lot of energy to release when you break them. And when I say break them, what I mean is, and this is, you'll see a picture on the next slide, but I'll start here. As you see, you know, there's three different phosphate groups. When I say break them, what I'm talking about is losing that last phosphate group. So then, instead of having adenosine triphosphate, which is 3, you have diphosphate, which is 2. So you're going to lose one, and that's how it gives up energy. Like I said, the next picture I'm going to show you makes that make more sense. But that will definitely be a test question. If you're going to make it very basic, like how does ATP work? The most basic answer is that it gives up one of its phosphate groups. It releases a phosphate group. It transfers a phosphate group, which we'll talk about later. If you just remember any of those, you'll be okay. Because I'm not going to have one question where all 3 of those are the options. Just remember, it's just giving up a phosphate group. The wording on the exam might be giving up a phosphate group, transferring a phosphate group, losing a phosphate group. I don't remember how it's going to be written, but that's how it's going to go. Any questions? All right. Let me make sure we're still recording. Good. Always good to see that. Anyway, so here's a picture of it giving up its phosphate group. Right here, you have the full ATP. Right, you've got the adenosine, your 3 phosphate groups. Here, we've lost one or gave one up, however you want to say it, which released energy, and now we're left with ATP. And I've seen this in so many different textbooks, obviously, because I've been doing this for a while. And almost every time you see it written in a textbook, the ATP is always like jagged like that, and the ATP is always smooth, because they really want to indicate that ATP is along with the energy, and the ATP doesn't have the energy. Well, doesn't have nearly as much energy anymore because it gave up its phosphate. So, any questions about this slide? And I do believe this picture is from the old textbook and not yours. So if you happen to look at it, if you're looking at your textbook and like, yeah, that picture doesn't make as much sense as the PowerPoint, you can download the PowerPoint to study that. So like I said, for the question on the exam, I might say it transfers a phosphate group, or it loses a phosphate group, or it gives up a phosphate group. For the exam, it doesn't matter as long as you can think along those lines, because I'm not going to get that specific, like have you memorize one or the other. But technically, and I guess your textbook doesn't mention this, but the old textbook did, so I'm going to mention it. It's actually transferring a phosphate group. Not that, again, I'm not going to get that detail for the exam. But it's not like that phosphate just disappears. What's happening is the ATP gives that phosphate group to something else. So yeah, that's just for you to know. It won't be on the exam. And honestly, I guess I could just leave it out. Even if you don't understand that, you can probably still understand the concepts that come later. But anyway, let's talk about it very quickly and give you some examples. Here's ATP helping a muscle move, right? The motor protein in a muscle. The ATP, you can see it gives up a phosphate group to the motor protein, which then links you with ATP and a phosphate group, and then it moves, and then those things are free to go off and they do something that we'll talk about later. And again, that's just an example. There are no questions about that. Or this. Here's one that we've talked about. We talked about not passive transport, but active transport, right? We talked about things going against the concentration gradient from one side of the cell membrane to the other, and how it requires energy to pump them against their concentration gradient. Well, here's an example of that, right? We have the transport protein. It's ATP has transferred its phosphate to it, which gives it the energy to allow it to push that solute against its concentration gradient. And the phosphate goes off and does its own thing. So does the ADP. Again, just an example. There'll be no questions about that on the exam. Here's an example where we're building a molecule. We're trying to make an XY. We have an X, we have a Y, but we need an XY. So we need energy to squish those things together. ATP gives it that energy by transferring a phosphate group to this X in this example. And then that gives it the energy for these things to come together. And then the phosphate goes its own way. The ATP also goes its own way. And we'll talk about what happens to that later as well. Also, not on the example, are there any questions? All right, this brings us to the ATP cycle, which is the easiest cycle you're going to learn in this semester. Also not in your textbook. But really, I don't know why it's not in your textbook, because without understanding it, you really don't understand how ATP works, despite how simple it is. So it's simple to teach, it's simple to understand. If you're really studying at the chemical level and the physics level, it's probably a little bit more complicated. But anyway, cells use ATP continuously. Specifically, the old textbook mentions that there's about 10 million ATP used and recycled each second in one working muscle cell. So imagine how many cells you have in your body. Then multiply that by 10 million per second. You're making, you're going through a lot of ATP. Of course, again, this is not your textbook, so that's a question I'm not going to ask you. And I don't normally like to give you numbers. That's very specific. But anyway, I'm just, in this case, I'm not going to ask you about it on the exam. But again, the ATP cycle is pretty simple. I've already given you half of it. All that stuff we were talking about previously, half of the cycle is the ATP giving up a phosphate group. And we also compared that to a battery, right? So this is like a full bar battery, right? ATP, it's got all three bars, so to speak, right? That's your fully charged battery. And then you use it. If you get a DP, right? I don't know how to draw that. It's not fully charged. Maybe it's only got, 2 bars. That's a horrible drawing. I'm no artist. But anyway, so again, ATP is your charged battery. You use energy from the battery. Then you have your dead battery, so to speak, your not charged battery. And then we go through a process, and that process is what we're going to talk a lot about. Mostly this chapter is going to be talking about going from ATP to ADP. But yeah, we go through this process to charge the battery, so to speak, and the cycle continues. Excuse me. So any questions about that side before I show you the next slide, which is just a different picture, a different version of this picture right here. Okay, then there we go. Next picture, same picture, just drawn a different way, right? You have a fully charged battery, ATP at the top. It releases a phosphate and an energy. That energy is used for work, so it's gone as far as we're concerned. Now we're just left with a phosphate and an ADP. Now we want to squish those things back together, right? We want to charge the battery. How do we do that? Cellular respiration. We use cellular respiration to put these things together. Then we have to charge back. And again, as much as we're barely talking about it so far, this is the majority of the exam for this chapter. Is going to be that one little thing right there is going to take up most of the questions. It's nice and easy when you just say it like that. Oh, how do we go from ADP and phosphate to a fully charged ATP? Cellular respiration. Nice and easy. But then it gets a little bit harder when we talk about how cellular respiration recharges the battery, so to speak. But yeah, again, I like to look at this as big picture to small picture, right? Big picture. You should definitely memorize this before you even worry about how cellular respiration works. Know that cellular respiration is what we use to make ATP. Okay, so the first thing I said today was make sure you know that cells need ATP for energy. If you forget everything else, remember that. That's the biggest thing you should get from this. The second biggest thing you should get from this is that it's cellular respiration that produces the ATP. And then once you have that memorized, then you should memorize this. Cellular respiration makes ATP in these 3 stages. You don't need to write them down right now if you don't want to, because we're going to talk about each one of them separately. But yeah, these are the 3 stages. This is, again, everybody's brain works differently. This is how I recommend studying it. Memorize it in the order in which I'm saying it, because if you memorize the big stuff, then you just add the details. Right? So the next detail to memorize is that cellular respiration is broken down into glycolysis, citric acid cycle, and electron transport. And then once you have that memorized, then you can start memorizing how each one of these works. But really quickly, those are not written in random order. Like sometimes I give you a list of things and they are in a random order. This is the specific order. This is how they go. They go on this stage, and you'll see why. The products of the glycolysis are used in citric acid cycle. And the products of both of those are used in electron transport. In the star, why did I do that? Okay, yeah. So your book uses the term oxidative phosphorylation, which is the technical term. But another term is called the electron transport. And for the most part, I'm going to use electron transport. And you'll see why. Because once you learn how it works, you'll understand why I call it that. And by me calling it that, I'm hoping it makes your exam easier. Because you're going to be like, oh yeah, well, clearly, like I said, a lot of the questions are going to be, in which 3 stages does this happen? And which of the 3 stages does that happen? So once I talk about electrons, specifically, certain parts of what they do, hopefully that'll kick something in your head. Like, oh yeah, electron transport. Back to the electron transport. So any questions about that? Okay. So to really quickly, just so you know, this, when I say this is not in your textbook, what I mean is I'm going out of order. So the information I'm giving you is in your textbook, other than, some nomenclature there. So the names are different. But what I'm teaching you is in the textbook, but I'm just doing it in a different order. Because what your old textbook did, what your textbook doesn't do, which I love, is it gives you an overview. It's like, and this is how I say this, I think is the best way to learn it. We're just going to get the big picture, and then once we have the big picture, then we're going to start going in and adding the details. And again, everyone learns different, but I think for most people, that is the best way to learn this, right? Learn the big picture, then start adding the details. So, that being said, let's move on and talk about it. You download the PowerPoint, you can watch this little animation about respiration. I actually haven't seen it in years, so I don't remember which part of respiration it is, if it's all 3. I can tell, well, we won't even get into it. Let's just move forward. You can watch it if you want. So again, this is an overview. You can take notes if you want to, but if you're a note taker, I would say you don't need to, because again, this is the big picture. Everything I'm about to show you now, we're going to come back and talk about in more detail. So again, if you're a note taker, you could just take this part off. But anyway, step 1, glycolysis. The most basic way to describe glycolysis is that we're using glucose and splitting it into 2 molecules of something called pyruvate. And I know what those stand for. I'm just going to go ahead and say, I'm going to put a cross through that. Usually this happens in the cytoplasm, i.e. not in the mitochondria. But as far as we're concerned, let's just say as far as we're concerned, it does not happen in the mitochondria. This is a stage does not happen in the mitochondria. That will be a test question. So it'll say which of the 3 stages happens in the mitochondria. Or maybe it'll say which of the 3 stages happens outside of the mitochondria. Forget. Anyway, yeah, this is one of those that does not happen in the mitochondria. We will talk a little bit more later about how glycolysis splits glucose into 2 molecules of pyruvate. But for now, just know that we start with glucose, we end with pyruvate. And even though we're going to add more details later, one of the details you're not going to need to know for the exam is how many molecules. Yes, we start with 1 molecule of glucose, and that produces 2 molecules of pyruvate. As far as I'm concerned, you just need to know that we start with glucose, we break it down, and we end up with pyruvate. Another word for pyruvate, which is what that little star is for, and you don't necessarily need to write this down, I just want you to know. Another word for it is pyruvic acid, and I mention that because I don't remember which term your textbook uses. But if you see pyruvic acid, it's the same thing. And I do this on purpose, and you'll see why. Because again, one of the test questions is going to be, I'll go ahead and tell you now, and then we'll talk about it later. One of the test questions will be, which of the three stages produces pyruvate? The answer is glycolysis. There's also going to be another question that says, which of the three stages is associated with acetic acid? And I can tell you now, for any question on respiration, if it has anything to do with acid, the answer is the citric acid cycle. And that's one of the reasons I don't want to call this pyruvate, or pyruvic acid. Then, if I call it pyruvate, I can tell you for the exam, if it says anything about acid, the answer is citric acid cycle. I made it easier for you that way. Always associate acid with citric acid cycle. Whether we're talking about citric acid or acetic acid or whatever, if it's acid, you should think of the citric acid cycle. Anyway, any questions about this big overview of glycolysis before we move forward and talk about a big overview of the next cycle? Excuse me, the next stage, which is the cycle, so the one we just talked about, citric acid cycle. Once again, with the nomenclature, another word for the citric acid cycle is the Krebs cycle with a K. But I do this on purpose to make things easier for you, because another reason I like to use the citric acid cycle instead of the Krebs cycle is because of all the C's. Citric acid cycle. And one of the test questions is to be which of the three stages produces carbon dioxide. So hopefully you associate all those C's together. That helps you remember. Okay, carbon dioxide, it's citric acid cycle, all the C's. And the cool thing about this, when we get to photosynthesis, it's going to be similar questions. Which stage uses this? Which stage produces that? One of the questions for photosynthesis will be, which of the two stages uses carbon dioxide? So whether we're talking about photosynthesis or respiration, when it's asking about carbon dioxide, the answer is always going to be some sort of cycle. In respiration, it's a citric acid cycle. In photosynthesis, it's the Calvin cycle. So again, all these C's, hopefully to help you remember, C's, carbon dioxide, cycles. I really hope that helps you in the exam. Anyway, yeah, that's like the big test question. There'll be other ones. We're going to talk a little bit more about how this works, but one of the biggest test questions is, which of the three stages produces carbon dioxide? The answer is citric acid cycle. And again, like I mentioned earlier, which of the three stages happens in the mitochondria? Well, this is one of them, right? Because you know the other one we talked about previously does not. This one does. Any questions about this slide? All right. Next word for attendance will be acid. The word is circle. So for those of you watching the video, not those of you online live right now, but ones of you who are watching this video on YouTube at a later time, and you take a screenshot, I should see a screenshot that has the word acid cycle. For those of you who are live online right now, just to reiterate, you're going to do what everyone else here has to do, which is send an email at the end with all the words and the accompanying screenshots. Of course, the one for the last attendance phrase isn't going to really make sense, but still need a screenshot. Anyway, not from you guys. So, big picture still here. Glycolysis and citric acid cycle generate a small amount of ATP. Matter of fact, all three of them do. So one of the test questions is going to be, which of the three stages produces ATP? The answer is all. But they produce much more ATP indirectly by transferring electrons from those molecules that they're breaking down, those food molecules, to something called NADH. I'll put it next to that. That will be important, just not right now. So maybe I should have included it. The point is, for right now, when we're looking at the overview, what I'm saying here is the electron transport chain, that's the one that's making the most ATP. That's like what it's all about. When we're talking about cellular respiration, it's all about the electron transport chain, that third and final part. But the other two are there basically to support it. And as the name indicates, electron transport chain, it's all about these electrons. So the first two, it's all about harvesting these high energy electrons from the food, so then we can bring them over to the electron transport chain where it can be used, or it uses it to produce ATP. And again, this is just big picture. We'll get into the details later. And like I said, yeah, I put it next to this, but this will be important later. You will need to know that NADH is the thing that's taking the electrons. And technically, once it accepts them, it's a different molecule. It's called NADH at that point. Anyway, then that NADH takes those electrons to the electron transport chain, and that's where it makes most of the ATP. The electron transport chain, oxidative phosphorylation. So I've used this term a couple times. I haven't explained it yet, and I still won't, but I mean, not right now. I will eventually, but I'm saying high energy electrons. These energies aren't just regular energy. These electrons are not just regular electrons. They're high energy. So we're stripping them from the food, first of all, in the first two stages. Then we take these high energy electrons to the electron transport chain, where that energy is stripped away, right? The electron transport chain uses that energy a little bit at a time until finally the electrons are accepted by the oxygen. So that'll be a test question too. Which of the three stages uses oxygen directly? Electron transport chain. And it just so happens when the oxygen accepts those electrons, it then forms a molecule of water, which will be another test question. Which of the three stages produces water? And the answer is electron transport chain. So the way that we're, keep in mind too, everything I'm saying, this is just an overview, which is part of why I'm going through it quickly, because we're going to go back and talk about all of this in detail. So the way this works is we have this mitochondria that we talked a little bit about, that we're going to talk about more later. And it has two membranes, like we mentioned earlier. And that inner membrane has a bunch of proteins embedded in it, electron transport chain proteins. They're embedded in it. So we've also talked about that, right? We've talked about proteins embedded in cellular membranes. And these things use that energy from the those high energy electrons, to pump hydrogen ions against their concentration gradient. So there we go, another term, another concept we just recently learned. Talking about active transport here, right? We are pushing things against their concentration gradient. Anyway, well, again, we're going to come back to that later and talk about this in greater detail. But another test question is that last bullet point. So one test question would be, which of the three stages produces ATP? All of them. Which of the three stages produces the most ATP? The answer is electron transport chain. So any questions about this slide? Again, big picture, the overall equation. You have glucose that's broken down in the presence of oxygen, and that gives us carbon dioxide and water and ATP. Now, there are some numbers, and I'm not sure yet if I'm going to get that detailed. If anything, you might need to know for every one glucose molecule, we get about 38 or 32, depending on the source you're looking at, 32 molecules of ATP. But I doubt that I'm going to get so specific and say, all right, for every one glucose molecule, we get one molecule of oxygen, and one molecule, or we use one molecule of oxygen, we produce one molecule of carbon dioxide, and we produce 6 molecules of water. I doubt I'll get that detailed. But actually, and I messed that up anyway. Sorry, still waking up. For every one molecule of glucose, we use 6 molecules of oxygen, produce 6 molecules of carbon dioxide, and produce 6 molecules of water. But the way that might help you remember it, I don't know, it helped me, just in case I do ask. Glucose is C6H12O6. So I just remember all those 6s. So I remember, all right, yeah, for one glucose, it's a big molecule, then 6 of everything else, other than the ATP. And that's it. We are done. Yeah, we won't even do the last word. So just send an email with all the words I've given you so far. One question about the material, and we will start at 9 a.m. And I'm going to get that lecture pulled up. So if you're online, what I'm going to do right now is end this. I'm going to hang up, so to speak. I'm going to end this call and then get right back on it. That way I can have 2 different recordings. One recording for today's lecture, and one recording for our lab. So if you're online, as I hang up, plan to be here online for lab instead of coming in, which for this one time, it's okay. And just come back in a few minutes. For the rest of you, feel free to stretch or get a drink or whatever you need to do. We got 10 minutes.